Okay, afternoon, everyone. And I did say afternoon and not good afternoon, so as to acknowledge the challenging times we find ourselves in. So we will all have to take that into account. And in spite of that, continue to do the good work that we are all doing. And good work is what my friend Stephen Chanock does a lot of. Stephen is a leading expert in the discovery of characterization of cancer susceptibility disorders in the human genome. He has received, unsurprisingly, numerous awards for his scientific contributions to understanding common inherited genetic variants associated with cancer risk and outcomes, including the Niehaus, Southworth, and Wiesenbach Award in Cancer Clinical Genetics. Some of you may also remember he was honored to receive the invitation for the Jeffrey Trent Lecture for our own institute. He's received the AACR ACS Award for Research Excellence in Cancer Epidemiology and Prevention. And he's elected member, a fellow pediatrician, I do want to point out. He's a fellow member of the Society for Pediatric Research, AACR, the Association of AAP, the Association of American Physicians, and the American Epidemiological Society, AACR, on and on and on, National Academy, AAAS. Steve's background is that he received his bachelor's degree, which I did not know this, in music from Princeton University, and his MD from Harvard, 1983. And he trained in pediatric infectious disease and hem-onc at Boston Children's and Dana-Farber, and served for 25 years, still serving? Okay, 25 years, 2.5 decades as a director of Camp Fantastic for kids with cancer. Stephen started here at NIH in 2001, where he's a tenured investigator in the genomic variation section of the pediatric oncology branch of the NCI Center for Cancer Research. And in 2001, he was appointed chief of the Cancer Genomics Research Laboratory, and 2007 as chief of the Laboratory of Translational Genomics within DCEG. Steve co-led the Cancer Genetic Markers of Susceptibility project, and from 2012 to 13, he was acting co-director of the NCI Center for Cancer Genomics and director, which he is now serving, of DCAG in 2013. So Stephen, please tell us about this great work you're doing on this important question.
It's a pleasure to be here. I have two corrections on that. One is I came in '91 as an investigator, and I first came to NIH in 1958. And I lived just down the street and learned to ride a bicycle and lived on this campus for a good part of my life. It's always a pleasure to come back to Building 50, which I know has NIAID on the top floor and NHGRI on the fifth, and I don't know who's on the other floors, but those are two very important floors in my life. It's a real pleasure to be here to talk about science, something that I'm not having a lot of chance to talk about recently, but I have actually new data for a paper that's out for review right now that I'll show towards the end. So those moments of being able to do science are far and few between, but they're very exciting and they keep me going and they get me up in the morning. So today I'm going to talk about epidemiology and genomics in individuals who survived or were exposed following the Chernobyl accident. And this is really I went the wrong way, sorry. Next, okay. Okay. This is a long-term commitment. In DCG, I became the director 12 years ago. Joe Fromani had run various versions of that for over 30 years. There's been over a 30-year commitment to understanding radiation epidemiology. And you can see people like Gil Beebe and Elaine Ron and Jeffrey Howe who started these studies. And we have very dear collaborators in two institutions. One is IEM, which is the Institute for Endocrinology and Metabolism, Mikhailo Tronko and Tatiana Bogdanova. And then Dmitry Bazika runs the NRCRM, which follows the cleanup workers. So we have a cohort of 29,000 people who were brought in during the Soviet age and cleaned up and then left, and we've followed them, knowing their dosimetry, exactly how much radiation they were exposed to and what kinds of conditions. And there's a long-standing tradition in DCG itself of individuals, and particularly the last person, Lindsay Morton, has really been instrumental in the studies that I'm going to talk about right now. So I think you all know about the Chernobyl power plant accident. April 26 at 1:26 in the morning, reactor 4 exploded in 1986. This was still Soviet Union, and it was the end of the Soviet Union, and this was a highly economically depressed area. And that's really important for the first story because the major source of nutrition for younger kids was milk from cows who fed in the fields that were contaminated. This was the major source of protein for younger kids. And so, unfortunately, the I-131 that gets spewed is concentrated in those areas. And as you can see here, just looking at the Ukrainian map, and Belarus got a very strong dose, as did parts of Russia. But the key thing here is really looking at the rates of thyroid cancer, which was the second most common cancer, probably the most, and I say probably is leukemia, but most of those individuals died rather quickly. So the time that it took to set up the infrastructure to be able to follow and capture and have the tumor repositories and the like, all were passed by. So the aplastic anemias and leukemias were indeed missed shortly after the accident. But thyroid is a very interesting organ that has the real propensity to develop after external beam radiation, radiation therapy, atomic bomb survivors. So you can see here that there's an age aspect. The younger you are when you're exposed, the greater the effect of whatever radiation you're getting. Like most things in pediatrics, once you hit adolescence, it's all downhill. But here, it's just the opposite, because once you're over adolescence, you're much less susceptible to this. If you're exposed as an 18-year-old, is very different than an 8-year-old in terms of the risk of thyroid cancer. Now, ionizing radiation is a class 1 IR carcinogen. And as I mentioned, leukemia and thyroid are the classic ones, but we know from therapeutic radiation given to patients that certainly sarcomas and other kinds of skin cancers can come up. Animal studies for decades have shown that double-strand DNA breakage with clean cuts, but no one has really understood what's the specific mechanism, how frequently, how does that happen or not happen. So that was really what's driven us to try and understand this, because at a public health level, it's a very important question of sort of, as we go from very high doses to lower doses, how much risk is retained as you go to lower doses, and at what point do you maybe not have risk for cancer? That's a big debate. And so by understanding an endpoint like cancer, which we also know, for instance, cardiovascular disease and particularly cataracts are very common in individuals who had exposure after the Chernobyl accident. And cataracts are probably the biggest problem as people get older. So papillary thyroid cancer has a higher incidence in women with, thank God, a greater than 95% survival. It's a cancer of young adults, highly differentiated, and the mutational burden is interestingly higher in older patients. So there's an age-related issue. And how much of this relates to what we know is the background, what we call clock mutations, signatures 1 and 5 that accumulate in any given cell. And mixed with that, I'm giving some of the punchline, is that this is a tumor that's driven by one driver, just one event. That's very unusual in cancers. When you look at melanoma or you look at smoking-driven lung cancer, there can be 7 or 8 separate drivers and subclones that are all unfortunately conspiring. So mutational analysis before had really shown that the BRAF 600 and TERT promoter mutations can actually be useful for therapeutic decisions. So we undertook this to try and understand thyroid carcinogenesis, insights into radiation-induced tumors. Is there a complex signature? Can we look at a tumor and say this is radiation-based or radiation-driven and this one is not? Because we know in the study that I'm going to show you that there is a background sporadic rate of thyroid cancer in young individuals, and the question is which ones are truly driven by radiation and which ones were going to happen no matter what, and I'll come back to that in the middle of the talk. And does the profiling suggest therapeutic steps? And how does this inform? This is sort of the controversial aspect of radiation epidemiology in the last 60 years, basically since the studies that really crystallized after following the survivors of the atomic blasts in Nagasaki and Hiroshima, which we study and are part of, and I'll come to it at the very end for a study, and we're doing a comparable study that Les has been very helpful with, is this issue of when you get to very low doses of radiation, there's some that argue that hormesis, that a little radiation is actually advantageous for a cell. There's some that say there's no effect, and some say it's not good. And there are lab models that support each of these three with different biases. But the question is, the clinical epidemiology is very difficult. So how do you really quantitate and capture people that live near nuclear power plants or radiation workers or radiation technologists, which we have big studies of following 40,000 people who have worked in radiation in radiology labs or radiology suites for the last 40 years. And this is an unanswered question, and this is one that really drives the public health questions. We know that thyroid, papillary thyroid cancer is very interesting from TCGA. In 2014, it was one of the first TCGA papers to be published. And here is what's called the classic commute plot. And this shows a lot of things all scrunched together. And the thing that's notable here is all of this is just the genetics. So over the objection of some of us, TCGA was done with virtually no real regard for what the exposures were and effort to be able to capture that, as well as the clinical outcomes. And we are spending millions of dollars trying to recapture retrospectively a lot of that information. And it's, how do I say, wholly, not in the religious sense, but filled with lots of missing data that makes it particularly difficult to be able to infer and understand things. Because we now know you can use somatic altered patterns, you're looking at mutational signatures, to work backwards in epidemiology. And sort of like NCIS, you can go to the scene of the crime, the cancer, and say, this person smoked, this person drank too much, this person was exposed to too much UV light or whatever. And whether that actually causes the cancer is another question that we'll get to. But as you can see here, what's important to realize is that about 70% of the patients had a point mutation, and they're just in 4 genes, BRAF, and then the 3 versions of RAS gene, H, K, and R, okay? And then about 15% had fusions, and they're relatively restricted. They're fusions in genes that are mapped to the MAP kinase pathway. And then about 25%, they couldn't find them. They did exome sequencing, not whole genome sequencing in the original paper. And then there were basically no copy numbers other than a little bit of 22q deletion. So we then decided that we wanted to comprehensively look at and really characterize the molecular profile in a cohort that had been identified and followed after the Chernobyl accident and put in what's called the Chernobyl Tissue Bank, which used to be in the UK. It's an international organization where each sample, each case is reviewed by 6 different pathologists, so you really have a common understanding of the phenotype. We've actually moved it to the NCI in the last couple of years. And this tissue bank had lots and lots of samples. And so I'm going to talk about the first 440 cases that we have access to. We have another 220 that are in the pipeline for papillary thyroid. And then there are other types of cancers like papillary carcinoma. And then there are also, adenomas that we're now looking at. So this is something that, the gift will keep on giving in our mind. So we looked at these 440 individuals who had fresh frozen, pathologically confirmed PTC, and we did whole genome sequencing, ending up with 374 cancers that met, the QC criteria. We did mRNA sequencing on a bit more, and then we had both non-tumor tissue from the other side of the thyroid, and then we had blood, and a few that are in these interesting trios that are allowing us to really explore what kinds of mutational events may be in the background. Now, the thing about, this particular study is we know exactly how much radiation each person, we have an incredibly good estimation of how much radiation each case got. We knew exactly where they were at the time of the accident, They were all diagnosed and had surgery in the same institution, and only 6 surgeons over 20 years have done that surgery according to certain protocols. And it's very, very methodical. So we have all these maps in Ukrainian where, taking this part of the thyroid out, this size, weigh this amount, and it's really, it's really unparalleled in the cancer world to have such exact, detailed pathologic information. And that's been helpful for some other things. And so within this 440, we were able to identify 81 individuals who were born more than 9 months after the accident from a region nearby. I don't have time to go into the population genetics, but this is all from the middle of the Ukraine. And if we look at the PCAs, this is an extremely homogeneous region that, all but 2 people are very, very similar in terms of their population genetics background. If you go to the east, where the war is raging on, they look more like the Muscovites. And if you go to the west, it's much more of a combination of the Poles and Lithuanians who for 600 or 700 years had large kingdoms that controlled a good part of the western part. So we have these individuals who had no exposure. So we, in a sense, have our, I wouldn't call controls, but our comparison group that's embedded within this. So 359 exposed with 250 milligray. Normally, when you get therapy, you get anywhere from 4 to 10 gray, so this is an order of magnitude lower. This is what people would get by environmental exposure or accidental exposure, and this is what is really worrisome in public health. If there were an accident or a dirty bomb or whatever, these are the ranges that people would potentially be exposed, and it's measured I-131, which was one of the main radionucleotides. It was released, and I-131, has a short half-life, so that we could really map out and know exactly how much. And the hard thing also is that I-131 is concentrated in the thyroid, because the thyroid uses iodine more than any other tissue in the body. So we know there's a particularly high dose to the thyroid per se. So we looked at a whole series of genomic variables that we created from the whole genome sequencing, the mRNA, microRNA, SNP, methylation profile, and telomere links with colleagues at the Broad and in our program. Danielle Cariati, who used to be here in NHGRI, come over. She's been terrific in this. And we looked at all the usual suspects, from single nucleotide variants to microsatellites to drivers, you name it. And then we have these wonderful radiation epidemiologists who have very, very sophisticated programs for taking the modeling of the radiation exposure and incorporating the uncertainty, because no one was measuring minute by minute, but with multiple measurements, what do we expect? And so the uncertainty principle is really important in this. So we did multivariable regression models adjusted for age, PTC, and sex. And so this is sort of what I've told the story in already one way, that the accident, the concentration in the fields, the cows drink it, the kids drink the milk, and they get what we estimate to be 20 breaks per genome, 20 per 1 gray exposure. Thyroid cancer, we know what that looks like, and it's these double-strand breaks are really important in generating the drivers, and that's what we're going to see in the next thing. So here is another commute plot of our 440, but this is organized in a way that no other one that's been published. If you look at the top, it's by environmental exposure. So the beauty of this, or the really interest, is the integration of epidemiology with genomics. So this isn't just saying, this is what we see when we sequence, and we'll figure out later whether someone smoked or not. So we know exactly how much radiation, and you can see from no exposure to 1 to 99 milligray, to 100 to over 500. And what's really striking is down on this line here, where I'm showing the red are the point mutations, but the higher the dose, the more the ratio shifts from point mutation to fusions. And the fusions are with just a handful of genes, all that have at least one of the participating members from the MAP kinase family or pathway. So it's really a very, very parsimonious kind of cancer. So when we looked first at, after taking the whole genome sequencing, it's one of the lowest in terms of mutational burden. the Chernobyl thyroid, lower than the adult, lower than just about anything else, 4 orders of magnitude less than what you see with smoking or UV, where there's just constant bombardment, and a lot of the mutations are due to the environmental exposures. Some of them may generate the mutations that are important for the cancer, but a lot of them are just spread across the background of the genome per se. And we see the MAP kinase pathway is what's hit almost exclusively, And then when we looked at the numbers and we were able to use the mRNA as well as the whole genome, we could find drivers in 98%, 93% were a single candidate driver. This is really unusual. There are very few pediatric cancers that are driven by one particular driver. Now we see that in the developmental ones. You see that in hepatoblastoma, you can see that in rhabdomyosarcoma, but not in Ewing's. You don't see it in osteosarcoma, ones that develop later in life per se. So it's a really interesting question developmentally that we want to look at and ask, why the thyroid? What is it about the thyroid? What's going on in the thyroid that you just need this one mutation and you get cancer? Whereas if you looked at all of us in this room and took skin biopsies, you could see many of these mutations, particularly BRAF, in the skin of just about everybody in this room who's had any UV exposure, but we all don't have melanoma. We all don't have basal cell carcinoma. So, to me, that's a really interesting next question that we're really trying to get at. So when we looked at the 176 fusion drivers, the RET, NTKR3, and BRAF, and PPARG, these are all things that fall into the MAP kinase, that are part of this one particular pathway that's very important for thyroid development. So now the next thing is we then decided to do a sophisticated analysis where we wanted to look at the EOR, that's the excess odds ratio. So what would you expect? What does radiation do? So this is where, as you can see, the rate of the EOR per 100 milligray against the dose that's exposed. And we could see a very strong effect. Almost all of these cancers are clonal. We don't see a lot of second and third clones per se. As I mentioned, there were a handful that had 2 drivers, and those, there you'd see quote unquote subclonality. We didn't see the 2 drivers necessarily on the same strand per se, and they would have, you could separate to the best of our knowledge that, and we'll come back when we talk about the metastases. Fusions primarily in MAP kinase, but again, these single clonal drivers are a hit at the wrong time. So the cut of the DNA is taking place, and if, particularly since fusions are overrepresented, the higher the dose of radiation, it's that single dose that hits and creates that particular driver. And this is something that really validated what there were lots of early reports in the late 90s of like 8 or 10 or 12 cases that were brought out of Belarus and out of Ukraine where they saw these RET-like mutations or fusion drivers, but they were really anecdotal. They were important, but we had no idea how and what breadth and what comprehensive aspect of that. So that's an important part of this story in our minds. So we then looked at the single base substitutions. Again, is there a radiation exposure signature? And the answer is no. We see the clock, the so-called signature 5, which we still don't understand the origin of, whereas 1 we know is the C to T, a very set, interesting, biochemical change. And otherwise, SBS8, which is unknown, which shows up in most other cancers as well, particularly pediatric cancer. So it raises an interesting developmental question. We then looked at the small events, anything more than 1 base, when you go to those, and we could see that ID8 and ID5 certainly showed up and were very strong insertions and deletions. So then when we said this question, all right, radiation-associated molecular characteristics. Mike Stratton and company had looked at 12 tumors of mixed types that were clearly driven by radiation, and they saw lots of inversions and deletions. Same time we published our paper, there was a paper from a German group looking at 190 post-radiation recurrent gliomas, that's kind of brain tumor. They saw the same thing, an increase in deletions more than inversions per se, again, of small size. And then in ours, we could clearly see that as well. And we could do this here. I'm showing the data normalized against the background of SNVs, sort of trying to take out the clock mutations because there are occasional inversions and deletions in the clock program per se, but they're a very small fraction. So, but we presented this as particularly a normalized fashion. So the structural variants, we could see these increased SVs with quite substantial, and they look to be mostly simple balanced SVs, okay? So in pediatrics, about 40% of the drivers that are fusions are very complex, where it's a very large part of a chromosome gets connected to a second, and there may be a third one attached to that, or that if they're just two parts of a chromosome or from two different chromosomes coming together, there's a deletion or insertion of hundreds to thousands of bases. Here, again, it's this notion of the clean cut that we could clearly see evidence for. So we can't see evidence for these complex or the simple unbalanced. And this was really important because then we said, all right, we have the fusion from the RNA, we have the DNA fully sequenced. We could go look at each and every one of the fusions and say, what does the characteristic look like? Will that help us to understand what particular mechanism is actually disrupted? And the answer is yes, we could do that. We could see enrichment for non-homologous end joining with the vast majority of the simple balanced having very, very small events around the way they would literally fit together. And so, knowing that there are multiple variations on the repair mechanisms, I'll run to that. But you could see for less than 20 base pairs of intervening loss, we had a high fraction had 4 or less bases, and there was a mixture of a few with 4 to 19, but that was really quite striking to see that. And when we looked at this again, having this opportunity of having the epidemiology and the dosimetry, knowing exactly, we also know, the incubation period from the time of all these people were exposed, April 26th to some point in May, and then when their cancers developed, or at least when they were diagnosed with their cancer as an endpoint and when the surgery took place, we could look at that and you could see that the microhomology analysis for what we saw in the less than 5 base pair events really correlated with the radiation. So with the more radiation, the more of these events are going on throughout the genome, but particularly they're important for those circumstances where they generate an actual driver. And then with this primary analysis, we know the double strand breaks are important and no radiation dose relationship for SNVs and SBS and driver genes, or particularly, we were surprised at transcriptomic and epigenomic profiles. I'm not going to go into that in detail, but we really couldn't see it. It's either RAS or RAF that drives what it looks like. So once the tumor starts, the tumor takes over, and if there was detritus of the radiation, it would be left behind. You wouldn't see it in the methylation pattern, and you certainly wouldn't see it in the expression pattern, which is a little bit different from what you see for instance, we have this big program in Sherlock lung where we're sequencing 2,000 non-smoking, and then there are about 5,000 in the literature of smoking, and you can see the residua of smoking in the RNA expression and in the methylation. Here, these are such parsimonious, hard-driving tumors, so to speak. Okay? So the question is coming back here, is there a way to determine whether there's a radiation characteristic or a set of those? So we then decided to look at specific tumors caused by radiation. Can we find reliable biomarkers? We could see these differences in gene rearrangements versus the sporadic in those tumors, yes, but that's not going to answer it all. We could see this from the Japanese data following the atomic blast, but not Fukushima. I'll come back to that a little bit later. And then we could sort of see how these different kinds of things really were laid out in the past here, okay, from previous studies that I'd mentioned. So the age, the tumor of initiation, the clonal clock. We know clonal clocks are going all through life in every cell per se. So the question is, can we use those to be able to identify with the mutations that we have and the insertion deletions, can we sort of develop a profile that would help us to say with greater and greater certainty that this is a radiation-induced versus a non-radiation-induced. The one thing I neglected to say earlier was the fusions are also more common the younger a patient, even in the sporadic cases. So if you go to the Philadelphia series of Andy Bauer and Amy Franco of 135 cases of PTC, the younger the child, the greater the likelihood they can have fusions. Not at the rates that we see necessarily in, post Chernobyl, but there is an age element to this, that if you're going to develop PTC and you're younger, there's a greater propensity to have a fusion driver as opposed to the BRAF or the RAS point mutations driving it. So here, we know that these events certainly can occur around at the time of, the tumor initiation. That's what we think of as driving the event due to radiation. But it's also plausible that some of these events, the radiation may have taken place, and then at a later time they're developing a cancer, and can we separate that? Or similarly, did they already have a cancer, and then the radiation occurs, and we're looking for, that was a sporadic case that was already started, and it just happens to be they were in, the Chernobyl region, and can we use these, these set of markers, particularly the clonal clock, 1 and 5, to be able to determine that? And the answer is yes, we can. So if we look at those individuals who have 2 breaks, less than 20 base pair gain or loss, what I mentioned before is really radiation related, we can see the age of the exposure in relationship to their clonal clock mutations and the accumulation. So this makes sense, so to speak. If we then go to the more complicated versions of mutations that are seen, and granted they're in smaller numbers, and particularly the BRAFs, we can't see the distribution of clonal clock in any way statistically lines up. In other words, as a regular accumulation, either the tumor, it was at the time of the tumor or just shortly before, within a year or so, that someone was starting that way, and the radiation may have actually induced that per se. So that's important. And I already mentioned the transcriptomic analysis, that we can see these things are all driven by the driver, not by the radiation dose. And similarly, the methylation. I forgot that I left those in. So I can't come to NHGRI and not talk about the germline for a minute. It won't be very long on this project, but when I get to the other projects, it'll all be about germline, okay? So don't worry, there will be germline discussion. So we looked at the PRS. Interestingly enough, thyroid is one of the diseases that has a very high heritability. So if you look at the GWAS, the genome-wide association studies for common variants, it's like number 3 out of 35 cancers that can be explained by SNPs per se. CLL and testicular cancer are the numbers 1 and 2, and multiple myeloma is not far behind. And there are others like esophageal squamous and bladder cancer that we can infer from our larger studies and projecting based on our large GWAS that we're not going to explain much of the heritability by SNPs per se. These are diseases that are going to be driven by other things. But the PRS is very powerful in thyroid cancer. And so what was interesting is we took the known PRS, there were only 12, and what was interesting is there's an inverse relationship. The PRS was lower in individuals who had more radiation. Now, this isn't statistically that exciting in my mind, but what it suggests is, to me, that the genetic burden could be important for the sporadic, but not for the PRS is not operating or in any way responsive to the radiation per se. Now, of these 440 individuals, we combed looking for any and every possible secondary finding, any possible gene that had been implicated of the 150 cancer predisposition genes, not to mention 400 other genes that have been implicated in radiation biology. You hear about them when I talk about clonal hematopoiesis in about 10 minutes. But we couldn't find germline mutations in any of these individuals. Granted, it's only 440 individuals. And we have another 300 that are in the pipeline right now. And with whole genome sequencing, it'll be interesting to see that. One of the other things that was really remarkable with the IEM group is just the care of the clinical care, and the follow-up allowed us to look at metastases. So the clinical annotation, all these patients are followed for life in this particular place. And I'll digress for a second, but when the war broke out in 22, there was a terrible disruption of the pharmacies in the Ukraine, and we have these contracts with our Ukrainians, and they follow, several thousand, particularly young women, middle-aged women with thyroid cancer, who'd had thyroid cancer, and they didn't have thyroxine. So we took out our credit cards, bought in Poland $9,500 worth of thyroxine twice and had it cross the border and go there so that for a period of time they had thyroxine. But it's a, you know, we have a very good relationship with them, and they're very good at following all of the details. And we could see that, you know, advanced disease is associated with larger tumor size and multifocality. But when we then had 47 METs available to us from the same individual, we were able to sequence them, and they look pretty much identical to the primary. There's one that wasn't, and that's because they're about 2 to 3% are multifocal. And the MET that we actually chose happened to be from a different primary. You know, there was a different primary that when we went and looked at it, we could at least see that particular driver mutation. But it's an interesting thing that when METs occur in the cervical region, they really are a spread of this particular tumor. So we don't see some of the things that people are interested in brain tumors. Interestingly enough, the METs are particularly, we see them with fusions more often than we see them with point mutations. And this is something we published last year. And we also know that the genomic architecture allows us to really see that the METs occur for different reasons. Sometimes there's a primary monoclonal that is metastatic monoclonal. Others that is primary monoclonal where the metastasis looks to be polyclonal, where in other words there's subclonality in the samples that we had. And then the primary polyclonal leading to a monoclonal spot somewhere else. We have a few instances of those, and we're following up on that right now. We have a fair amount of sequencing going forward on that. So our priorities are to go deeper into the characterization of radiation and PTC characteristics. We have another 215 FFPEs and another 100 fresh frozens that are in various stages of our pipeline analysis. And we're working closely with the groups in Toronto and Philadelphia about this age issue, because this is, we can see a strong radiation effect, but we can't say that age is not a contributing factor to this, and we, you know, have to be realistic about that. So single cell RNA ATAC is going on right now, because one of the things we're really interested in is why and what's the vulnerability of the thyroid? Why this one mutation? Why is radiation hitting there and doing that? Whereas there are many other organs that get radiation and don't necessarily go on to sort of monogenic somatic mutations that are driving particular cancers. So we are also looking at somatic alterations in the normal tissue with double-stranded sequencing with, right now with Ludmila Alexandrov out in UCSD. And then we've just started with a number of centers looking at those kids who got radiation therapy. The older pediatricians will remember when the kids with ALL would get treated with craniospinal 1,200 or 1,800 rads as prophylaxis against CNS disease. That's gone its own way. We also have, we're just finishing a study of about 150 individuals with PTC from the World Trade Center cohorts in New York, where we don't know of radiation of I-131, but again, as being a significant issue per se. It was so hot that some people think that there were different radionucleotides, and there is some debate about that. But we've jumped in to at least do the genomic characterization to see, do we see features that would help someone sort of epidemiologically think backwards on this, and be asking the question of what would be the driving force in that. So that's it for the PTC, and particularly Lindsay and Danielle and my former OxCam graduate student, Eric Dawson, who, you know, now at NVIDIA making obscene amounts of money programming things and coming back and helping us. I'm going to talk about something he's helped us, you know, out of the goodness of his heart. I mean, he's a wonderful OxCam. I know, Elaine, you used to run that, so he's one of, we've had 3 or 4 terrific OxCam people. So the next thing I want to talk about is, in this setting, we designed a family study to address this question of transgenerational effects of protracted radiation exposure following the Chernobyl accident. So this is a study. We wanted to look at de novo variants. Sorry, I thought I corrected all the slides. less, because we had this conversation in the fall, and this one I missed, I'm sorry. De novo, should be de novo variants, not de novo mutations, in adult children born to liquidators and evacuees. So we had a family design, which we published in the American Journal of Epidemiology. So we've now collected over 290 families, but the first paper that we published 4 years ago was 105 families with 130 children, all born more than 1 calendar year after they left the Chernobyl area. We know exactly how much the mothers and or fathers were exposed. We did short read, very high coverage, 80x. We didn't want to miss anything. And now we have the 290 with a clonal hematopoiesis study in progress I'll talk about, but we've now extended this a bit more. We know de novo variants are very important for risk for a number of pediatric diseases. Autism certainly one of the top there, but certainly some of the catastrophic pediatric developmental disorders. And certainly the Icelanders have been out front in developing some of the pipelines. It's an essential type of variant that due to random mutations for the most part. Now there are these very interesting group about select genes where there may be a higher predisposition for a mutation per se, but that's again a very small fraction of that. But these variants are generated by a classical pipeline, and that's how the Icelanders and how Ben Solomon, when he was with Wendy Wong and company over at Inova, did it, and would see between 40 and 100 that would increase with the age at conception. Newly appearing variants are known as de novo variants, and to do this you have to see both the parents and then say, oh, this only exists in the child, and that's a lot of QC activity. And it's a critical step in evolution because this is how new variants are introduced and can be selected in populations or deleted per se. And particularly when you think about all of the the mutational signatures and primary pediatric diseases that we see, de novo variants are really important. In pediatric cancer, it may be that 25% of bone tumors, osteosarcoma, are due to de novo variants arising in families with very strong pathogenic mutations. All right. Prior data on the transgenerational effect due to radiation certainly has been there with animal studies where they would get 2 to 4 gray, which is a pretty high dose, and evidence of DNA repair, non-homologous end joining, with some structural and chromosomal events. And we know that FISH is used in the commercial world for following people that work in the nuclear regulatory, in nuclear power plants, that they have blood drawn periodically. And if they have, there are like 6 chromosomes, if they, by FISH, have rearrangements in those, then they're considered to have been exposed too much and either stop or they pause for a period of time. But there have been no real good human studies using whole genome sequencing, particularly for lower protracted dose exposure, and particularly the ability to look at microsatellites. There was originally a paper of Nagasaki of 3 families, highly anecdotal, very interesting, and we'll come back to that. So when we looked at our 105 families, we could see the distribution followed exactly what had been published in all the other papers, from Denmark, from Virginia, from Iceland, and with the real favoring of a C to T, which is, really the hallmark of mutational signature 1, which occurs most frequently because of the biochemical thing. But we were able, because we had the radiation exposure, to look at the age of the parents, obviously, and saw that the paternal effect is very strong and the maternal was just borderline overall. The radiation, we did 6 different ways. I'm just showing you the one that seems to make most of the radiation epidemiologists happiest. That's what we put in the primary table. But in the paper, there are 5 other ways in the supplements. We presented it with 3, and of course, reviewers wanted 2 other ways, and we put them in, It's always, you want to have a happy family when you do that. And we looked at whether smoking or alcohol was also part of this, and we did not see that. So there was no difference in mutational distribution between the Chernobyl trios and population-based studies. When we looked at the different types of events, from clusters and complexes to indels to microsatellites, SNVs, we couldn't see any of those subcategories, so to speak, of genetic events enriched due to radiation exposure. They all had the paternal effect to varying degrees. And we were too small in this study, and we've been corresponding with the Icelanders about sort of asking the question, is that paternal effect more important for microsatellites versus SNVs per se? And microsatellites have gotten more interesting. But when you can look at the parent of origin estimates based on flanking SNPs per se, we could actually see now the maternal effect. And we know that about 42% of the DNVs we can assign to either mother or father. And the more recent study just about to come out of Iceland suggests that 18 to 20% of the events are maternal. And this is, this is the old shibboleth that, the spermatogenesis is going on continually, whereas the eggs are basically set at the time of birth. Well, there is obviously some turnover if there is these kinds of events taking place. And so it raises a really fundamental, interesting question that I don't have an answer for. So we had done this by going through the maternal, paternal, child, going through a series of QC steps. And we actually reviewed every single one. We looked at the IGV of all of them. So Meredith Yeager and Mike Dean looked at hundreds of them, excluding some, and they each did it independently, not knowing the radiation, and then crossed their data, and they were about 96, 97% congruent between the two of them, which is reassuring for looking at this kind of data. So we then have now turned this into a pipeline that we're just about to publish, and we've done this on a different study I'm not going to talk about right now, a smaller study in Brazil. There was an accident where this basically this vessel with cesium dropped off the back of a truck in a relatively poor part of a city, and a number of people came and picked it up, and children played with this. And so we've been sent a number of those families, and we're doing the same kind of study. And so we use the new pipeline, and what we're able to do is it helps us to be able to identify indels and microsatellites at a higher rate per se. So we've gone back and we've done this on our Chernobyl 105, and we see no difference. Statistically, radiation is not, in our minds, associated with a higher risk for any of these kinds of events. It's just we now, particularly the microsatellites we can get. And as people are now starting to use PacBio sequencing, and we're going to be doing that on some of the families where we see some very complex things, just to be able to better sort of pull those apart. We will do that. But we're very excited that this is now more automated than it was before, because the 105 families took Mike and Meredith literally 4 or 5 months of just continually looking at IGVs. And that, boy, I'd be cross-eyed if I did that. But God bless them for doing that. So our study considerations, I think it's important to realize that we've looked at peripheral blood. And this is in adult children. So there is a survivor bias, but not much of one. I mean, again, we're looking at parents who had doses that were less, by and large, less than 1 gray, extended over a period of time. So we didn't have parents who had leukemia or aplastic anemia because they got 4 or 5 gray and went to Moscow and died within a short period of time, but conceived a family beforehand. So there is a bias, but this is an important public health message for particularly Japan, and I'll come to that in a minute. So we're moving forward with this, and we can see that the risk of deleterious DNVs is probably very small, if at all. I mean, if there is something, if there is a pediatric developmental thing, it's probably more random as opposed to radiation-driven. We had adequate power to stratify substantive increases in DNVs in adult children, and we've done epigenetics as well, and we really don't see much in the way of what happens in the family. The doses were lower and extended. And this is important because when we put our paper into Science, there was one reviewer who insisted that we put one sentence in, and he or she wouldn't allow it to go by until we said there's an interesting alteration in the balance between new gonadal DNVs and DNA repair. And this may be, there may have been some repair taking place. And it's an interesting question. Animal models, this is a highly debated issue. So since then, in Britain, they looked at 60 families who were nuclear test veterans who had been in some of the islands in the Bikini Atoll, et cetera, for the British testing, and they saw no increase in DNMs. And this has been really an important study. Once we publish this, I still go to AACR, I get multiple requests for interviews from the Japanese press because of Fukushima. Because remember, Fukushima had doses that are about 1/10 of what these individuals in the Chernobyl saw. And what's interesting, though, is one of the major reasons that people have not moved back into Fukushima, which is now 12, 13 years ago, is parents of, or individuals of potential parenthood were worried about mutational events and didn't want to go back and have children because they would have mutations. And this is something that's really been hard to convince the Japanese, because the right-wing press seized upon this, and it's really hard to dislodge that. The timing of the radiation could be critical, and we have more families that don't really show a significant change. We're just sort of on the final stage. Long-read NGS of highest and lower doses is going on, and I already mentioned the Brazil, but we have this big study that I've been trying for 12 years at RARF, and God bless Les to actually go there and assure them that they can have secondary return of results, which is a really hard thing for the Japanese to get their hands around. But there's a study of 580 families where they're sequencing 60x, and it will be very interesting. There's different radiation fallout from that, and the exposure curves look a bit different, but it will be very interesting to see this. And they're in the process of doing their first sequencing. We're looking at some of the QC of that. So we see this as really a very important kind of study. So the last thing I'm going to talk about for just about 3 or 4 minutes is acquired somatic mutations, clonal hematopoiesis and chromosomal mosaicisms, two ends of the extreme. I mean, we were the first out of GWAS to show the chromosomal aberrations back in 2010, and they occurred about 0.5 to 2% of the population. Boy, sorry. I'm trying to go back in time. Sorry. And what I wanted to say is that we knew that inherited environmental factors increase, particularly clonal hematopoiesis. And so Kelly Bolton, another terrific graduate of the Oxcam who was in my program 10 years ago, is one of the leaders of clonal hematopoiesis, has been instrumental in these studies. She had published a study looking in Nature Genetics, the Sloan Kettering experience, and seen increase in clonal hematopoiesis of indeterminate potential in the expected genes, but increased in individuals who'd either received radiation therapy or high therapeutic doses. So in other words, these very strong environmental exposures increase the risk of CHIP. So we wanted to know at lower doses, as you would expect in Chernobyl, can we see that? So we went to our liquidators, and we have a target of over 2,000 that we're doing this, but we started with these 290 families of the mothers and fathers. We also have the children, and the objective was to look at the frequencies and types of CH, and does environmental radiation exposure increase either the CHIPs or the mosaic chromosomal aberrations? And are they risk factors for cardiovascular disease, cataracts, and diabetes? Because we follow these cohorts, and we can look at non-cancer outcomes, and that's something that as the numbers get larger, we'll be able to do. So we've now done 581 of the target 2,000, higher doses in the fathers than the mothers. And this is 1,000x of 620 genes, all the expected known CH genes, and anything ever implicated in radiation biology we put on that list, wondering if any of them would do this. And then we also did the GSA version 3 chip. So the association here, we have red marrow doses, okay, which is different. Dosimetry has to think about different body parts getting different kinds of radiation exposure. And in this study, we've done the red marrow dose. And what's interesting is we don't see any real effect of the red marrow dose in having an increase or decrease by EOR so far with our first 580 individuals. I'm presenting the fathers separately. The mothers, they have lower doses because by and large, 90% of the liquidators were men who were basically conscripts sent in by the Soviet government to go clean up, not told much, not trained well, and had extensive exposure, whole body exposure. So this is of particular interest to us. So when we looked at the children, we saw really no appreciable CH or MCA. And we think this is informative because there really haven't been studies of young adults and children for clonal hematopoiesis. And the big question in clonal hematopoiesis, are these events early and they're suppressed because you were really good at holding back something? Or is it because your DNA repair as you get older starts falling apart and you generate them later in life? So the fact that we don't see anything with 1,000x coverage on, 330 30-year-olds to speak of is interesting, and we're going to continue to do this. And that's an important sort of pediatric observation. I'm not saying it either makes or breaks the story, but it's an important thing to fit into our trying to understand how and why clonal hematopoiesis emerges in what age group and to what degree. So having the 1,000x gives us those VAFs, okay? So for this work, certainly Meredith Yeager and Mitch McKeela and Amy have been marvelous, but many others. And I'll finish by just saying that here in DCG, we do wonderful training, and we have 100, 120 fellows at any time, and I guess there are questions. And DCG does lots of exciting things. We love to collaborate, okay?
I don't have any online questions yet, so if there's questions in the room, please come to the microphone. I would have predicted that CHIP was not going to be responsive to the low radiation. Was that your prediction also?
Yes, but we need to do a sufficiently large number. Just like our transgenerational, there are people who say, well, why are you doing this if you don't see the effect? This is where genomics and public health really do intersect. We need to know this because there is lots of low-dose radiation exposure, and there may be more, either planned or unplanned, and we need to know what that really means for subsequent generations. But I agree. I didn't expect to see a whole lot. I wondered whether we'd see some different genes per se, as opposed to the usual, methylation, DNA repair, p53, TET2, AXL. I was surprised that we didn't see anything but what has already been described in the adult population.
Thank you.
Very cool. Very cool, actually, about getting the synthroid to them. That's really neat. What are you doing with secondary findings in the Ukrainians? How in the world are those being followed up?
Well, it's an interesting question. So we started the study like in 2017, 2018, and the IRB there viewed this as a purely research finding. So we're having new conversations about this because at least the NCI's position is if it's an IRB and it's a truly research study, then the return of secondary results is not necessarily required, okay? And this is hotly debated in different places, I realize. And the Ukrainian, and we're talking about, unless you and I may not go to the Ukraine for a while. Once it settles down, we may go. I mean, I go periodically, but I haven't been since 2020. And I have no intention of going until I know the missiles are not firing. But I think it is an important question. The Japanese are clearly doing this because they're doing it in a clinical way. Their IRB protocol is telling the patients you're going to get this back, whereas in the Ukraine it's much more post-Soviet. How much information you get, what you give back is much more restricted. I mean, we have studies in Belarus where they don't tell them anything, and we say we'd like you to be able to share the overall results of thyroid, and now we have a big breast cancer. Women who are exposed to the Chernobyl accident seem to be having an increase in breast cancer because lactating breasts take up a lot of iodine, and so parchment women at that time could be a real risk. They won't allow us, and they won't go back and tell anyone that. I mean, it's a very different world. But I think we will get to the point of return of secondary results, but it'll take some time.
I do have another question that's
Yeah.
completely different. So given that there are some experts in cancer in dogs and radiation therapy. I wonder how have you been
Oh, we've
combining
talked to,
your results?
Elaine has come talk to us.
The
and-
Chernobyl dogs.
Yeah,
and the-
exactly. And absolutely. I mean, the Chernobyl wild dogs are very interesting because their survivorship and the generations are so short. You know, we're now looking, what, 6 or 8 generations out? 15, sorry. Okay, yeah. So the question is that transgenerational effect would have been selected. It would be hard to know what the de novos are at this point. Dogs have lower de novo rates, which is an interesting thing. I learned that relatively recently. Someone wrote me a question at Purdue. What the wonderful woman? Debbie Knapp, yeah. Okay. Hi, Paul.
Very nice talk. So I have a question about the chip, that part of study.
Yeah.
So you mentioned those don't have chip, but have you looked at overall mutation burden in those patients? I mean, in those
Well,
individuals.
we're looking
Not
at
only the chip
we're
genes.
looking at 620 genes with targeted sequencing
Right.
1000x.
Right.
We don't. I mean, in terms of having background like
Right.
synonymous variants, etc.
Well, yeah.
It's a great question. I don't know the answer. They may have already generated that, but we haven't talked about it in our data. In the paper we sent in, we didn't talk about synonymous, and like I showed you with.
Or in other genes, not
Yeah.
in those 600 genes.
Well, but those are the only ones that we have 1000x coverage. We have 80x coverage in 440 cases of thyroid cancer, and then we're getting data from a couple of population geneticists of 500 or 600 whole genomes across Ukraine, just because they're interested in looking at some interesting questions of the population genetics and could the radiation be related to L1s and things like that.
Right, because the context is radiation may cause overall mutation increase,
Yeah.
but their selection, just as you said, these are younger individuals, so they may be selected against.
I mean, we don't see a lot of point mutations. That's the thing. The SNV load is very low. I mean, it's basically clock background mutation that you would see in any normal population at basically that rate. Another question?
Yes, I just have a brief question on how you defined adult children for the study, what the cutoff was.
They're 30 years old and older, basically.
older.
Because the study, you left between 87 and 92, and we started recruiting this entire study in 2017. And so the latest anyone had a child, I think the youngest was 28 or 29. So adult children from these family trios, but not kids. As a pediatrician, let me make that very clear. Sorry if I misspoke.
It's all right. I get it now. Thank you. My other question is, I know your focus is on thyroid cancer, but what I'm interested in, why not other cancer, right? For example, some of these children were exposed at really young age, right? But it's interesting they're not other types of cancer.
Correct. Now, so after the accident, and it was seen pretty quickly based on sort of old Israeli and Israeli data from giving kids with seborrheic dermatitis radiation to the head, and then the ALL experience of craniospinal prophylaxis, that there was a focus on thyroid. So we have a bias in that the IEM only was looking at cases that a number of them were screened multiple times or followed. There were 3 different cohorts focused on thyroid. Now, the background rate of pediatric cancer is far too low to have anything appreciable show up other than anecdotally 1 or 2 in these kinds of numbers. There was no sort of pediatric cohort set up after that. As I mentioned, there were a number of kids who had ALL and aplastic anemia. Some of them were sent to Moscow, and some of them never made it out of Pripyat or that area, just because it's 1986 and the medical system was pretty Spartan, and this is the Soviet Union falling apart.
Thank you.
Great, thanks everyone, I appreciate that, and we'll take
I'm
a few more
here to
questions
meet
afterwards.
Yeah, meeting
Awesome.
with
Thank our
trainees. Yeah.
speaker.